Hello, everyone, and welcome to Lunch Break Science. I'm Ariel Johnson from the Leakey Foundation, a nonprofit supporting human evolution research and sharing discoveries about what it means to be human and more. Happy New Year, and welcome to our first episode of 2025. Today, we explore the evolution of gibbons, fossil identification, and much more. We love hearing where you're joining us from, so be sure to let us know in the comments. And if you enjoy staying up to date on human evolution research, like, subscribe, turn on notifications, and support our work. Every action helps us bring you more amazing content. Joining us today are Leakey Foundation grantees Christopher Gilbert and Alejandra Ortiz. Chris, thank you. Chris is joining from New York and is a professor of anthropology at Hunter College and a research associate at the American Museum of Natural History. Chris searches for ancient primate fossils in the Neogene deposits of the Shivala Hills in India and works on ape and monkey evolution in Africa. Alejandra is joining us from Peru and is a research scientist at NYU. She's a biological anthropologist specializing in ape and human evolution with research experience in Peru, England, France, Germany, Tanzania, South Africa, and Kenya. Her work focuses on human skeletal biology, paleoanthropology, dental anthropology, and growth and development. Let's welcome Chris and Alejandra.
Thanks a lot for having us.
Thanks so much
for being here.
Great to be here.
Well, we're so excited to have you and hear about gibbons and your recent publication. So, what is it that makes the evolution of gibbons so fascinating?
Yeah, it's one of the most interesting questions, really, in all of primate evolution. I think the first fossil apes appeared in Africa around 25 million years ago, and there's many fossil apes that are known from that time onwards. They're actually more common than monkeys for many millions of years. We also know from the molecular clock that lesser apes, or gibbons in other words, separated from great apes around 20 million years ago. And yet among all the fossil apes that are known, and again, there's a whole slew of different species, there's no agreed upon fossil gibbons until you get to some of these fossils in China that are 7 to 8 million years ago. So there's a really large gap in time and space in the fossil record. Fossil gibbons must be around starting around 20 million years ago, and we know they got to Asia at some point, but how and when that happened is virtually unknown because we have no fossil gibbons between 20 million and 8 million years ago. And they're gibbons. They're very distinctive animals. So it's like I said, really interesting question.
And something about gibbons I find fascinating is their great diversity. So including the Siamang, there are like 4 living hylobated genera and up to 22 species compared to the great apes that are only represented by 7 species. And this is kind of like paradoxical, right? Because like as Chris mentioned, like the fossil record of gibbons is, as of now, like very, very limited. Also, if we pay close attention to gibbon evolution, you might be able to note some sort of like resemblances with our own hominin lineage. So for example, in addition to species diversity, hybridization between species is also possible. And also like both lineages, so hominins and hylobatids, like show reduced like sexual dimorphism among other things.
So, Chris, you were last on Lunch Break Science in 2022. What have you found since then?
So when we were last on, I think you'd asked what I hope to find in the future. And I said we hope to find more fossils of Kapi. And unfortunately, I can't say that we found any more of Kapi just yet. But that's probably to be expected given the rarity of primates and given the fact that collection of a fossil have been taking place in the Shivaliks for almost 200 years systematically, and Kapi is the only specimen of its kind that's ever been found. So unfortunately, I can't tell you that we have more of Kapi just yet, but we do have more interesting primates. So from the site here of Taplal, we have been fortunate enough to find a couple of Shivapithecus specimens that are actually was really I think a fun story. If we go to in 2018. Yeah, my colleague Primjit Singh found this partial tooth. It's like 2/3, almost 3/4 of a tooth, and we recognize it's a tooth of the great ape Shivapithecus, which is the early relative of the orangutan. And so that was great. And then about 5 years later, my colleague Rajiv, we're about in the same place, and he found this little fragment of a tooth, and he said immediately, he's like, I think this goes back on Primjit's tooth. And I was like, well, what are the odds that 5 years apart you find the other little tiny fragment? I thought, well, show me a picture when you get back to the lab. And sure enough, he got back to the lab, and if we can go back and forth real quick, you'll see here. And then it fit right on. It was exactly the other part of the tooth. So now we have this very nice complete tooth. It's like an upper M2 or M3 of a Schiopithecus. In addition, we also found another specimen. And just again, these don't seem like a whole lot of specimens, but I just again repeat that primates are really rare. So we're very fortunate to find this is actually a pretty decent haul. And we also found in blue there, you see the outline of this proximal. It's a part of a finger bone. And my colleague, Dr. Biren Patel, is pretty convinced it's probably digit 2 or 5. So your index finger, your pinky finger. And it's the first postcranial fossil of an ape known from Ramnagar in the 100 years that people have been collecting there. So not a whole very large quantity, but certainly some interesting specimens, and we continue to find interesting primates. And probably some of the most interesting things we've actually found are some rodent specimens that actually are very useful for giving us hints about age, because rodent species are only around for a few 100,000 years. So if you find one, you know that you have to be within a few 100,000 years. So those help us figure out how old everything is. So I'll come back to that a little bit later. But that's what we found since we last talked.
It's just amazing to find, 2 parts of a tooth years and years apart. I mean,
Yeah.
it's not the only time that's happened, but it's just like, again, like you say, what are the odds?
Yeah. It was pretty fun. Like I said, when Rajiv sent me the picture, it was I had to just kind of shake my head and chuckle, because what are the odds?
So, how do you identify a fossil like Kapi? And how does dental morphology help distinguish species?
Right. That's a great question. This is why, paleontologists are trained in anatomy or osteology in general. So fossils broadly, are usually identifiable versus rocks because they have a clear anatomical shape. So if you know what the ends of bones look like, they have a very distinctive shape. So you can recognize that versus sort of jagged shaped rocks. Some of the most recognizable fossils are teeth, which are covered in enamel, which when it preserves is very hard and it's very shiny. So it has a certain sort of, very clear glint and shine to it that sort of catches your eye. And again, if you spend a lot of time studying anatomy, you recognize teeth versus rocks based on anatomical landmarks and things. And teeth are, kind of like gold in the fossil record. They're some of the most useful things for telling apart different groups of animals. They're one of the most differentiated parts of the body. So you can identify pretty broad groups of animals pretty easily. If you're spending time on a farm and looking at farm animals, if you've ever looked at it like a pig versus a horse versus a cat or a dog, their teeth, or a cow, they all look very different. Similarly, a primate tooth is very different from an antelope or a rhino or an elephant or giraffe. So when we found Kapi, when we found that little tooth, we knew it was a primate because it was distinctive. It clearly wasn't any other broad groups. Getting it down to the species level or the genus level is much more difficult. It can be hard to identify through teeth alone. So figuring out exactly what kind of primate can be tough and often needs more careful study and a nice comparative sample. So when we first found the fossil, we thought it might be some sort of small ape. We knew it wasn't a monkey because monkeys have distinctive teeth, and we knew it wasn't Schiopithecus because it wasn't big enough. So we thought it was probably looked like some sort of ape, and that's why we turned to Alejandra, who's done a lot of work on living ape teeth, in particular gibbons and other apes as well.
And with that, you might also get a sense that, like, as Chris mentioned, like tooth shape, function, and evolution are all interconnected. So the teeth of primates that have like a relatively similar diet compared to those who have like different ecological niches and evolutionary history have like similar tooth morphology. So for example, it will be relatively easy to differentiate like the dentition of like a small insectivorous like primates such as a tarsier that you see on the top of like left side of the screen from like say a great ape such as a gorilla. Or it will be relatively straightforward to differentiate a cercopithecod monkey from like that of a siamang. So for example, monkey molars like are kind of like high crowned with two cusps on each side and two parallel ridges that round from side to side. So meaning from cheek to tongue side. Hominoids, on the other hand, have like molars with kind of like low, rounded, like conical cusps arranged in a kind of like a Y-shaped pattern. But the key question here is, what do we do when we try to identify specimens within the hominoid lineage? Or what do we do when we're dealing with stem hominoids or stem catarrhines that are less derived than present-day monkeys? And this is where like things like get trickier. But one option is to rely on comparative analysis in the lab. This is a way more boring task than being in the field, but arguably as equally important. Earlier methods like were generally based on like linear measurements, and actually like the way we image our targeted sample has changed radically over the past like 10 or 15 years or so. So we moved from like caliper measurements to 2D images in which every single tooth that we had or that we wanted to image was like we needed to make sure that the lens of the camera, like the skill and tooth position were leveled and placed in a standardized way. So it was a very boring and time-consuming like task. And we have moved from that to 3D surface scanners. And micro-CT technology has also become more widely available and affordable. So, and this is great because it allows us to look at the internal architecture of like biological systems in a non-destructive way. We will talk more about the methods that we use to determine Kapi's like affinities in a minute during the presentation. And also I will be happy to talk more about like some of the pros and cons of each method during the Q&A section as well.
Well, thank you so much. I really cannot wait to jump in. But first, I do want to take a moment to thank our series sponsors, the Anna Gordon Getty Foundation, Camilla and George Smith, the Joan and Arnold Travis Education Fund, and viewers like you. We want to give a special shout out to our live viewers, Nancy, Darren, Rohit, Majo, LVJ, Chris, Catherine, Barbara, Rutger, Jill, Lou, Jennifer, Chris, and Gabriella. It's fun to see so many grantees watching. So again, just thank you. And Pete, we will miss you today, but we'll see you next, or we'll see you live next month. If you want to help us create more episodes of Lunch Break Science by sponsoring an episode, contact us at production@leakyfoundation.org. And you can also help us by making more episodes by donating through YouTube, giving, or the link in the live chat. Now, let's turn the virtual floor over to Alejandra and Chris and dig deeper into gibbon evolution.
Okay. So I just thought we just sort of go through, what our recent publication is about looking in more detail at gibbon evolution through the fossil Kapi ramnagarensis, which we first described back in 2020. So just a quick reminder. So we're in the Shivalics, the Shivalik Hills. This is the Shivalik group of rocks, which goes across northern India and Nepal and into Pakistan on the Palwar Plateau. And these rocks have been producing fossils that have been of interest to scientists going back, at least 200, probably longer than that. But systematic collection began almost 200 years ago. A lot of classic localities, some of the earliest fossil apes known are from these areas of the Shivaliks. In particular, a lot of classic areas in Pakistan and around here in India, a place called Haratayanga. There have also been fossil apes known for the past 100 years from the place called Ramnagar, which is where we're working, which is near the town of Jammu up in northern India. And all of the kind of areas, most of the fossiliferous places here are basically just south and southeast of the town of Ramnagar. So here's a close-up Google Earth view just to give you a sense. The town of Ramnagar is is my cursor? Can you see that on the screen, or is that not there? If not, no problem. You can see the town of Ramnagar sort of in the upper left-hand corner there. And the way that the geology works, if you look all the way to your right, in the right lower corner, you see Kulwanta and the mountain Ramchun Peak. Those are the oldest parts of the deposits. And then things as you move to the north, to the northwest, get younger as you move to the northwest. So we've been able, through a lot of really hard geological work, mostly by a Baldwin fellow at the Leakey Foundation, Deepak Chaudhary, and also my colleague Rajiv Patnaik and Chris Camposano and Dan Pepe have been working on the geology. So we've been able to sort out like the stratigraphy here, and we've been collecting data for sampling to do paleomagnetic analyses to figure out how old everything is. But the important thing here is that we know that the very same level we have where Kapi was found here at Sunater, which I'll show you in just a second, is at the same level. So same age as far as we can tell as where we have some Shivapithecus fossils. So this is what the area looks like where we actually found Kapi. It's kind of amazing because it's really not a terribly productive area, actually. I mean, there's not that many fossils we found there, but 2 of them have been primates. And this is back in 2015. This is an expedition that was funded by the Leakey Foundation. And we've been looking all morning, and I was kind of halfway up the hill, and I sat down for a second just to take a quick break and kind of maybe take a GPS point. And I looked down to the side, and like I was just talking a second ago, saw sort of a glint, a little bit of a shine in the dirt. And so I kind of dug around, and out popped this little tooth. And as soon as we found it, we knew it wasn't a monkey and it wasn't Shivapithecus. And Rajiv said immediately, "That's a primate." And I said, "Yeah." And we kind of got excited because it struck us as looking a little bit like a gibbon. And like we just explained a few minutes ago, however, there's no gibbon fossils known until 7 or 8 million. So I thought, well, what are the odds that we're finding like, the oldest gibbon in the fossil record? I thought, well, not very good. So this must be something else. So we got home, got back to the lab, and started looking around trying to figure out what this thing was. And that's when sort of we got it CT scanned. So there's a nice image here we could study in more detail. Sometimes it's hard to see stuff with the enamel, just how shiny it is. And so we started coming across Alejandra's work, who did all this work on all these sort of younger fossil gibbons. And we thought to ourselves, if we're going to figure out what this thing is, we're going to need Alejandra's help. So I'll turn the floor over to her now, and she can tell you what she thought when we sent once we sent her pictures of this thing.
So first, I wanted to say that comparative anatomists and evolutionary morphologists generally spend many, many years of their lives visiting museums and around the world collecting data, in my case, on hominoids. But before moving on, I wanted to also give kind of a shout out to Terry Harrison, Dr. Terry Harrison, who introduced me to the wonderful world of gibbons when I was a PhD student at NYU. It was actually Terry who put Bunopithecus on my radar. And what initially started kind of like a side project, became kind of like a big component of my research agenda. And it is thanks to that Bunopithecus paper that we demonstrated in a way that we can indeed identify and separate, as you can see on the slide, like a gibbon taxa based on tooth anatomy alone. And it is also thanks to that paper that I became involved in many, many other cool gibbon projects, such as the one led by Chris. So, and I remember, I feel kind of old at this point, but science does take kind of a long time. So, and I remember that I was a postdoc student, at ASU when Chris contacted me. And when he first sent me this photo of Kapi, I could note some resemblances to gibbons, but I also had at the back of my head this idea that there isn't much we can do with just like one single like tooth, with just one single molar. But since I already had like a bunch of like comparative data, I thought, okay, why not? Why not being part of like Chris's project? And as some of you know, like qualitative features like can be very informative, but also kind of like subjective in a way. So we decided to run a more quantitative assessment through a technique called like geometric morphometrics, where you take point or landmarks at key anatomical features to analyze like shape in a more objective way. And that's actually what we did for Kapi. So, and Chris, can you move to the next slide, please?
Yep.
So, and to analyze Kapi, we collected a sample of as many extant ape and Miocene catarrhine lower M3s as we could, and did shape analysis to capture like cusp position and the basic overall outline of the tooth. And to my surprise, because I was trying like very hard to convince myself that it was not a gibbon, the resulting PCA like separated gibbons from all major groups of like stem catarrhines and stem apes. Like with like if you go to the right part of like the plot, like PCA separating, I'm sorry, to the left part, like you have like PC1 negative that shows kind of like relatively shorter, like broader, like M3s with more horizontally aligned, like cusps, like the mesial cusps, the ones that are like at the front of our mouth. They also have like a more centrally placed, what we call hypoconulate, which is like the distal, like central cusp that we have at the back of our molars, and more overall peripherally placed cusps. Then we have to differentiate that from like elongated M3s that are like in this plot, like on the right part, like PC1 positive, that have like mesially positioned like protoconates, a more like buccally positioned like hypoconulate, so like the distal cusp at the center, and more centrally overall located like cusps. So in summary, like Kapi plotted well, like inside the gibbon distribution, as you can see, like based on this plot, as did other like fossil gibbons that are like were better known as Bunopithecus and Euanmopithecus as well.
Right. So we became pretty convinced then. I mean, it was a gibbon. That's what we published it as in 2020. So in other words, if we look at an overall just schematic of sort of living monkeys and apes here, we placed it, right here as a very close relative, early relative of living gibbons. So these are all apes are sort of from all living apes are from this point forward. And then so we were a bit surprised that a couple of years ago there was a paper that came out by actually Terry Harrison and Chinese group, and they sort of basically said that we got it wrong. And they said, no, Kapi is instead one of these early sort of, it's not even an ape. As you see here, their sort of idea was that it's something that branched off before the common ancestor of living African and Asian monkeys and apes. So in other words, something that's sort of like between living South American monkeys and living apes, but something that's not even an ape, and it's this group called Pliopithecus. So when they said that, I'll be honest, my first reaction was, oh shoot, what did we screw up? How do we what do we do? And then so you start looking at a little bit more detail, and they had a whole lot of very specific ideas that they put forth to say why Kapi was not a gibbon and why it was instead this early primitive non-ape. And when you looked at all the things that they said it has relatively smaller part of the tooth here, or different position, part of the tooth there. All these things, there wasn't any quantitative data to actually support that. So sort of, if I can bring up a quote from like the Big Lebowski, it was just like, well that's just like your opinion, man. So we decided, this is how science works. You put forward ideas, and then other people have to go test them. So we said, well, let's test these. If these are true, we can test these with data. And again, Alejandra is the one with all the data on all these teeth and things. So I'll let her tell you exactly sort of what we did to test these various hypotheses put forth by this paper by Gibbons.
So as Chris mentioned, we decided to reanalyze or analyze Kapi's molar again, like paying particular attention to like all those features that, according to like G and colleagues, made Kapi a pliopithecoid or like a stem catarrhine rather than a hylobated. So for example, we calculated relative areas of like the 5 like major cusps by tracing the tooth outlined and the main fissures like dividing the cusps, like something that you see on the bottom, right bottom part of like your screen. And also to quantify like the relative position of like the 2 cusps, like closer to the front of our mouth, like meaning what dental anatomists refer to as protoconid and metaconid. Like we calculated the angles between these cusps and a centrally located point as a kind of like as a baseline at the anterior front end of like the tooth. So based on those like cusps or like landmarks, we took like the ratio of like these like 2 angles like 1 and 2. And when the protoconid angle, so like this, I don't know if you can see my cursor, like this one here, is greater than the metaconid angle, like the other one, like the value of the ratio, like is greater than 1. And that means that like the protoconid is mesial or like more forward, like relative to the metaconid. All that, like I'm trying not to be like super technical, even if probably I am more technical than I wanted. But that kind of like supported our idea and kind of like were in disagreement with like those like opinions that were put forward in this Joan Mopithecus paper. So when the metaconid angle like is greater, like the value is less than 1, which means that like the metaconid is more like mesial or like, again, like closer to the anterior front of the tooth. So an angle of like 1 like indicates that both cusps that are like on the front part of like the tooth or anterior part of the tooth are like transversely or like more horizontally aligned.
So yeah, so that we collected all this data, Alejandra did all these measurements, and then so we looked at these things then in sort of turn, looking in detail to see if these sort of things were true. So first, they said that they had relatively smaller cusps at the back of the tooth. So again, Alejandra measured all of those cusps. It actually calculated areas of them all. So we plotted them. And we have on the next 2 slides, I'm not going to go in too much detail. I don't want to bore everybody to tears, but we have, Gibbons are in green here. And the group that the other research group was claiming they were in is an aqua here. And they were claiming that, again, that in this particular feature, that Kapi was smaller than Gibbons. It was distinct. But if you plot it, it plots well within the distribution of Gibbons for this particular cusp size. It was the same thing when we looked at the other cusp size that they said it was smaller than. It actually is well within the normal range of variation of what is known among Gibbons. So it wasn't distinct in any of these ways that they were claiming that we could actually go ahead and measure. And then similarly, we looked at some features on the outside of the tooth that I'll show you in CT scan. So this is our Kapi is the gray one, and below it is the accepted. We all accept this is a fossil gibbon, this fossil Yuan Mupithecus from China, and they had argued, if you can see the shelf at the top, which is being highlighted in red there. They suggested it was longer and more extensive in Kapi, and as you can see on the far right, you'll see that highlighted in red. You'll see actually Yuan Mupithecus has a longer, more extensive shelf on the outside of the tooth than Kapi does. So that wasn't true either. They also sort of made the idea that this crest here that's highlighted in red in the lower half of the screen, they said that was oblique. It wasn't straight. And as you'll see, actually, Yuan Mupithecus below it is really more oblique. It's got like a big crink in the actual a tooth compared to Kapi. So again, that didn't really hold up either when you actually compare them next to each other. And then finally, they suggested that there was evidence of this very distinctive feature that you find this early group of non-apes. You find this triangle, which again I've highlighted in red on the right side. The Kapi is the gray one. Below it, in brown, there is an example of an Indian non-ape Aplypithecoid, and they have this distinctive triangle in the middle of the tooth. And they suggested that that triangle was at least partially present. And I've blown up at the bottom right part of the screen. I've blown up Kapi as big as you can get it, so you can see there are no crests. There is no triangle. So in every way we could actually go and compare, and that's and actually test these with actual data and look and test these hypotheses. We found none of the assertions that were being made to distinguish Kapi from Gibbons was actually true. And then Alejandra was able to do a multivariate analysis again. So go ahead, Alejandra.
And if you remember the original PCA from like the first paper that we published on Kapi, you might recall that there was a great amount of like overlap in hominid and hylobated like shape space. So there was a lot of overlap between like living hylobated and like living great apes. We know, however, that there are like some kind of like obvious size differences between like hylobated teeth and those of great apes and several stem hominoids actually. So in our revised assessment of Kapi, we decided to investigate like size and shape. So as opposed to only shape as we did in the past, such that we could get, hopefully, or that was our hope, like a greater separation among groups and calculate the probability that Kapi belongs to like any of the taxonomic groups like that we included in the analysis. One of the most common techniques to address like such questions is discriminant function analysis. So in our discriminant functional analysis, like Kapi was treated as an unknown like case, which means that Kapi's like group membership was calculated a posteriori, like assuming like equal like prior probabilities like across groups. Okay, so what did we get? So our results, like as hopefully I'm trying to like convince you, like, or if you can see, like we're kind of like compelling. Around like 90% of specimens in our sample were correctly classified at a group level. So that means that a Dendropithecoid was classified as a Dendropithecoid, like a Pliopithecod was classified as a Pliopithecod, and so on and so forth for like 90% of like the time. And again, like Kapi, which is in this like red star here, plotted exclusively within the hylobated distribution and was classified as hylobated with around like 89% posterior probability. And equally important, like considering like the G and colleagues paper is that Kapi was, so they were claiming like this paper, like G and colleagues were claiming that Kapi was a derived Pliopithecod. But actually in our analysis, we found that only 0.8% like of posterior probability that Kapi was classified as a Pliopithecod. So I think like our like results were like super strong, like regarding Kapi being hylobated and not being for sure like a Pliopithecod as they were claiming.
Right. So, big picture, I think what it means is still kind of what we sort of, originally sort of said back in 2020, and that is that it's really Kapi is still the most compelling evidence of for the earliest fossil gibbon that we know of. It's no matter how we analyze it using quantitative data, that is clearly the most likely possibility at this point. And what it means, and what this figure is illustrating is we have, like I said in the beginning, fossil apes. Primitive fossil apes are in Africa around 20 million years ago. We know Yuanmupithecus is in China at 8. And so having Kapi in India right around 12 and a half, 13, it's really filling both a geographical, it's intermediate, almost it's intermediate morphological, anatomically as well. It's intermediate geographically, and it's intermediate in terms of chronologically. And so I think what you're catching is sort of your, a little bit of a snapshot or window into the evolution dispersal of gibbons into Asia. Obviously we'll test that with additional fossils, but the cool part about it is that since some of the geological work we've done since then we know that Shivapithecus, which is a fossil great ape, a relative of orangutans, they show up the earliest known when they show up is at the same level as Kapi. So it seems to be, it's not just lesser apes coming in, but probably great apes and lesser apes. It's probably whatever was in the environment allowing, ape dispersal to take place, which makes sense. You're probably getting both great apes and lesser apes are sort of a part of this evolutionary dispersal event, kind of making it into Asia. And you're catching sort of the first wave in the Shivalics as they're coming through around 12.5 to 13 million years ago. And so that's really what the paper has to say. And so I think Ariel, I'll turn it back over to you.
Great. So thank you for giving us a exciting glimpse into your paper. We have added a link to the article in the comments if you're interested to learn more. Behind every great discovery is a dedicated team. Tell us more about the incredible group of people who contributed to the research and this publication.
Right. Actually, most of our team is actually in the field right now, finding fossils and working on the geology. So, Rajiv is our co-PI. That's Primjit. Primjit has been working with us for a long time. Of course, has found Shivapithecus specimens as well as a lot of great work on fossil rodents. Rajiv is co-PI and co-director of the project in India. He's been working the Shivalics for most of his career and found numerous specimens of Shivapithecus, as well as other important primates and other fossils. The other co-authors of our project in the foreground there is Dr. Biren Patel, who's also co-PI, and he's really a founder of the project. He basically there's Biren as well, bugged our co-author John Flegel to send us to India way back when we were postdocs. Here's the geology team. Deepak on the left is a Baldwin Fellow, Chris Campuzano, Dan Pepe, Rajiv. They've been doing really amazing work on the geology and helping us sort out exactly how old and sorting out the stratigraphy at Ramnagar, which is really important. John and Kelsey, who's not pictured here, were the other co-authors on the paper. I just want to mention them real quick. They helped us identify the fossil and run phylogenetic analyses as well. So yeah, everybody, it's really is a total team and group effort. I also want to thank, I know Rohit's watching, Rohit and Wasim, also other Leakey Baldwin fellows who also worked with us and have done important fossil finding and continue to do important work. It's a long-term collaboration, and we're having a lot of fun, a lot of great success finding fossils in the Shivalics and elsewhere.
Well, it's great to hear, about how many people are involved. It's definitely a group effort.
Yes, 100%. Exactly. So, yeah.
And I want to give a shout out to more of our live viewers. Apologies if I mispronounce any names. Brad, Michael, John, Ida, Nancy, Mike, Tam, Kyle, Joanna, Fikru, Jose, Kimambo, Zepezi, and Marco. Thanks for joining us live. We cannot wait to answer your questions, which is going to come very next. If you haven't gotten your questions into the chat yet, now is the time. We will take our first question from Rutger. Do you have a hypothesis about the earliest origins of gibbons? For example, are they living descendants from early Miocene small apes from Africa, or do they have a closer affinity with larger European apes and shrunk to a smaller size during their evolution? Rutger is a grantee as well. So it's always fun to get scientist questions. Okay, so take it away.
Yeah, I mean, what do you think, Rutger? I mean, I feel like I should ask you. I would, my own. I don't know. I'll let Alejandra speak for herself, but I would guess that they're probably from from some of those small apes or some of those fossils in Africa somewhere. It's possible even there's something that is, we just can't recognize it because it's just not derived enough yet. But that's my guess rather than a a dwarfing of any known European ape that I'm aware of. Alejandra.
I totally agree with you, Chris. Actually, like one of my dreams, like eventually when I have time, is to like look again at all like the different like hominoids that are like in the hominin vault in Kenya. And I mean, I wouldn't be surprised that there is like a gibbon like over there that we haven't identified as such.
Well, thank you for your question, Rucker. Our next question comes from Darren. If you were able to extract DNA from something this old, how similar would it be to current day gibbons?
Good question. Right, because they've actually been able to get not DNA but proteins and stuff out of some specimens of fossil orangutan relatives like Gigantopithecus, which is super cool. And we've actually sent some of our fossils of Chilodapids to actually have some of these proteins taken out as well. So that would be interesting. So as far as, I mean, I would just expect it basically that whatever you would find would be more similar to gibbons than to other living apes, which would be great apes. And that's really as far as you'd be able, I think, to go with it. I don't know that exactly. Not being a molecular biologist, I wouldn't be able to tell you like the percentage of genes or ACTs and Gs that all line up, but basically you would just expect that it would come out as being more similar genetically to living gibbons than to great apes.
Our next question comes from Chris. This isn't a primary question, but have any bird fossils been found in the same formation as Kapi?
Yeah, very good question. I don't recall any specific bird fossils that we have found in the past 10 to 15 years. However, since I certainly believe that in the Chevalics more broadly that there are bird fossils. That probably I have to check with Rajiv if he's found any at some point, but I'd have to. I'm not familiar enough with the bird fossil record to say specifically, but my guess is in the Chevalics broadly, yes. Specifically at Ramnagar, have we found any recently? No, but it's also, people have been collecting them for 100 years. So again, it's quite possible. I'm just not familiar enough with that section of the record. My apologies. I should know, but I don't.
So our next question comes from Diane, who asks, why are there so few gibbon fossils? Is it an issue of preservation?
That's a very good question. I think probably preservation could be part of it. I mean, at least in this part of the world, again, primates are like 1% or less than 1% of the fossils that you find. If you find enough fossils, you will eventually find some apes, and they just seem to be extremely rare. I don't know. The current diversity of gibbons might just be a recent thing. Rather, so in the past they just might have been much less common relative to great apes than they are today. Alejandra, do you have any thoughts on that?
Yeah, one of the reasons I think like it's very hard to find like a fossil gibbon is that it's looking at their habitat. So like they live in forested habitats. So like in forested habitats, like it's very hard actually preservation. It's very hard for like fossils or like for like specimens, biological systems to preserve like well. So that's one thing. And if we compare it with like great apes, and this is like a pure assumption, but my other feeling is that like they are like overall like way more gracile. So which means that probably like preservation, it's even harder for like such a gracile like bone or like a specimen compared to the great apes. But I would say that most likely it has to do primarily with like the habitat that and the forested habitat and preservation.
Yeah, there's a similar issue actually with monkeys. The whole guenons, which are the most speciose group of living monkeys in Africa, are actually there's very little of a fossil record there as well. It's a similar issue is what's thought is that they specialize in tropical forests that don't do well in terms of preserving fossils a lot of, and they do seem to be a more recent radiation, which is why they can all interbreed still and all that kind of stuff. So living gibbons is probably a combination of kind of all those things as well.
Let's see. Our next question comes from Francis, who asks, what other species do you both work with?
I did my dissertation on fossil monkeys, so I have a lot of experience with fossil monkeys. I also collect in Wyoming very early primates, so early relatives of lemurs and tarsiers and things like that. So I sort of and study monkeys, and really I find all of primate evolution. So I've at some point kind of looked at most radiations of primates at some point. Alejandra?
I actually started with modern humans only. I'm native from Peru. I'm a Peruvian native, so like there is a lot of like archaeological material related to modern human. But then I fell in love with like apes as well. So I basically work on like hominoids, and so I think it's a good kind of like match or like combination of skill sets. Like Chris like does more like monkeys and other like non-hominoid primates, and I do like hominoids more often, I would say. And then right now I'm also working on macaques, but living macaques from like Cayo Santiago.
Oh, it's great to have such a background to bring, especially when you're doing comparative work. So our next question comes from Robert, who asks, what are the most common fossil apes and what are the most common fossil primates?
The most common fossil apes. Really, probably the most common. The most common fossil apes are really the ones that probably branched off. It depends on what time period what you were talking about, but early on, it's really a group of primitive apes that are sort of branched off before probably gibbons and certainly before great apes. There's actually not a whole lot of fossil great apes, and there's not a whole lot of fossil gibbons, but there's a whole lot of fossil apes. Most of them are these primitive apes that branched off probably before all of the living the common ancestor of the living apes. And then the second part of the question was, again, I'm sorry, it was most common fossil primates. The most common fossil primates. Geez.
Colobles, paracolobles? Like I don't know. There are plenty of them.
Yeah,
but I don't know if there's another species
I don't know.
that has more.
Yeah, I don't know what one genus or like group it would be the most. Again, it probably depends on what time period we're talking about.
Yeah.
So early on in the 50 million years ago, if you go out to Wyoming with me in the summer, you're almost guaranteed to find a few fossils of Neotharctids, so early lemur relatives and early tarsier relatives. They're actually some of the more common fossils of mammals at that time. If you're in the Fayum in the late Eocene, early Oligocene, you find very early primitive monkeys are again pretty common. In the early Miocene, again, you have a lot of fossil apes, and then as you get to later Miocene and sort of onwards, actually monkeys are really common. And in fact, probably one of the most common ones is actually this giant monkey, this relative, so the living gelada baboon, is one of the most common fossils you find. In all the sites where you find hominins and everything else, one of the most common, the most common primates is, if not the most common primate, is Therapithecus. This a giant relative of the living gelada. They're extremely common. So that would be one of the more common ones.
We had a really great episode about Terapithecus a few years ago featuring Dagmohit Getahun.
Yeah.
Definitely, well, I'll have to add that link to the chat because that was a wonderful episode. They're just such amazing fossils.
Yes.
Okay, let's get to our next question from let's see here. Actually, I'm not quite sure who it's from. It'll be surprised. Okay, from Darren. It's my understanding that we separated from chimps around 6 to 7 million years ago. Would we have separated from gibbons prior to the age of this fossil?
Yes. So we great apes, including us, and gibbons, lesser apes, share a common ancestor going back around 20 million years ago. It would be when we last would have shared a common ancestor.
So we have one final question before we get to that question. I just want to ask if you have any advice for anyone starting out in science or wanting to pursue a career in science.
Just contact your professors and ask if you can get involved in their research. Is probably the best thing you could do, and that leads to oftentimes opportunities studying in the lab or even getting to go out to the field. Alejandra?
Kind of the same thing. Try to find as many or take as many opportunities as possible, in order to get training and in field work, lab, and some techniques, and also probably be resilient, stubborn. And if this is what you find if this is your passion, just follow your passion, even if you're gonna have lots of failures down the road.
Well, I'm just going to give a quick shout out to more of our live viewers. Again, it's so great that we have, all of you watching live and contributing questions. But a shout out to Rohit, Tyler, Majo, Julie, Hannah, Edward, and Stephanie. Now we will take our final question of the episode, and that comes from Tam, who asks, "What are your dream fossil finds for the future?"
I'm still waiting to find the partial or complete skeleton of Kapi, so we can confirm that it is indeed a gibbon. That would be great. Or really, I think anytime you find something new is always really exciting. Alejandra?
Same thing, probably a more complete skeleton or at least dentition of Kapi. And also I'm very interested in the late Miocene period. So when they're the chimpanzee lineage and the hominin lineage diverge. So hopefully we'll find soon some fossils that were on that kind of time period right there, when we both split from each other, chimp and hominin lineage.
Well, thank you so much for joining us today. Unfortunately, that's all the time we have for questions. And thank you for submitting such great and just complex questions. Chris and Alejandra, thank you so much for joining us.
Thanks again.
Well, thank you all for taking a break from your day and feeding your brain with the Leaky Foundation. Until next time, stay hungry for knowledge. Lunch Break Science is brought to you by the Leaky Foundation and made possible by the generous support of the Ann and Gordon Getty Foundation, Camilla and George Smith, and the Joan and Arnold Travis Education Fund, as well as viewers like you. Show your support of Lunch Break Science by subscribing to our channel, clicking on notifications, and giving us a thumbs up, or making a donation to help us create new content. Still craving science and can't wait for the next episode? You can feast on the Leaky Foundation's content library with past episodes, lectures, our podcast origin stories, and more. Thank you all for tuning in, and see you next time.